Hi everyone. Today's sets of notes will look at the impact of mutations on the different pathogens that we've been studying this year. So the two main types of pathogens we've been looking at have obviously been bacteria and viruses. So this dot point is relating to how mutations in both of these different pathogens can impact on how challenging it can be for humans to treat different diseases caused by these. We're going to specifically look at bacterial resistance as well as antigenic drift and antigenic shift in viruses. So let's take a closer look at those now. We're starting off with mutations in bacteria. So this one's fairly straightforward and links closely with what we spoke about in our topic of evolution. So bacteria obviously can develop a resistance to antibiotics, and antibiotics are our main form of drug that we use to treat a bacterial infection to help our second and third line of defense obviously combat that bacteria. So what exactly is happening when mutations occur in bacteria? Well, we know that bacteria replicate via binary fission, and that theoretically every daughter bacteria cell should be identical to that parent bacterial cell. But just like in replication of human cells, there can sometimes be mutations that pop up in the replication of bacterial cells. And those mutations can then obviously lead to different traits in the bacteria. So this is all falling under the umbrella of natural selection, which we've looked at in our evolution topic. So natural selection is at work here, where there will be variation that exists within a bacterial population because mutations have occurred and therefore created new strains of bacteria that have different traits to the original one. If that bacterial population is exposed to a selective pressure, for example, a particular antibiotic, those bacterial cells that have the desirable mutation, so the antibiotic resistance, are obviously going to be more fit to survive, be able to survive, and then potentially pass that trait onto any future bacteria that they form via binary fission. Okay, so why is that a problem for us as humans? Well, if bacteria do develop a resistance to antibiotics, it means that we can't use that particular drug to treat the bacterial infection and give that assistance to our second and third line of defense. This can obviously create complications depending on what type of bacteria has infected an individual and where in the body it has infected it. So for example, if we're looking at the bacteria that causes pneumonia in your lungs, there are strains of pneumonia that are antibiotic resistant, and there are certain antibiotics that we cannot use to treat that infection. So that might potentially mean that we need to look for a different type of antibiotic to help us treat that bacterial infection in the lungs, or we need to be able to make our patients more comfortable so that we can wait for their third line of defense to kick in and take care of the bacterial infection themselves. When we're looking at mutations in viruses, they're a little bit trickier. So we obviously know that viruses are a non-cellular pathogen and they do require a host to be able to replicate. We also know that all viruses have either DNA or RNA within their protein shell, their capsid. And that's what we're looking at the image at the bottom of the screen here. So this is just a 3D representation of a virus particle here where we've got our nucleic acid, the RNA, the green stuff in the middle. We've got that brown sort of circular coating, which is representing the capsid. And then on that capsid, we can see some red, blue, and purple proteins. So those red, blue, and purple proteins, they would be acting as antigens for this particular virus particle. And some of those proteins do have specific names. So you can see on the screen there that I've labeled the red ones as neuraminidase and the blue ones as hemagglutinin. You don't need to know too much about these, but if you do want to investigate them further, most virus particles will have some version of neuraminidase or hemagglutinin on their surface, because these are the proteins or the antigens that virus particles will use to help them enter or infect a cell, and then also proteins that they need to exit that cell successfully and then spread those virus particles to other cells too. The reason that I've mentioned them on the screen here is when we do look at mutations popping up in viruses, it's a little bit easier to notice those viral mutations when we look at the names that scientists give to certain strains of viruses. So really common viruses where mutations pop up all the time are things like the influenza virus. And the way that scientists name an influenza virus is they actually look at the different antigens or the different versions of those antigens that are on the surface of this influenza virus. So for example, they might look at the one on the screen here and identify that the neuraminidase and the hemagglutinin antigens that are present on this cell, we've got hemagglutinin version 3 and neuraminidase version 2. Okay, so when they are looking at this cell, they will then name this, when they're looking at this virus, they will then name this virus the H3N2 influenza virus, so that all scientists can then recognize that this strain of influenza, this strain of the flu virus, has these particular antigens on the surface. And they're obviously going to be different to a different strain of influenza or a different virus that has been named in a slightly different way. It might be H5N7. Okay, so it's indicating that there are different antigens on the surface of these virus particles, and therefore our body will recognize them differently. So why are we looking at these different antigens on the surface of a virus? Well, if a mutation was to happen in a virus, and it is usually very common for these mutations to spontaneously pop up, particularly in viruses that are formed from RNA rather than DNA, purely because DNA usually has a lot of self-checking mechanisms to prevent mutations popping up. RNA doesn't have those self-checking mechanisms, so when a mutation does pop up, there is nothing to correct it. So if we were looking at something, for example, like the influenza virus, which is an RNA virus, we know that there are many, many different strains of the flu virus because we get vaccinated against them every year. If it was the same strain of the flu virus infecting humans every year, there would be no need to get a new flu vaccine every year because we should have already created memory cells in our initial vaccination to that strain of the flu. But no, we are encouraged to get a new flu vaccine every year. And the reason we're encouraged to get a new flu vaccine every year is because the influenza virus, the flu virus, undergoes a process known as antigenic drift. And that's just a fancy way of describing that every year flu viruses will experience spontaneous mutations. And those spontaneous mutations that occur in the RNA of this influenza virus sometimes control the shape of the antigens that will appear on the virus capsid. Okay, so it might be a mutation that is changing the shape of the neuraminidase antigen, or it might be a mutation that pops up that changes the shape of the hemagglutinin antigen. Okay, and if that happens, it means that that new virus particle that is released from the host cell when it's ready to move on and infect a new cell is actually going to have different surface antigens. That means that if your body has already created antibodies to the original shaped antigen on the virus particle, those antibodies and memory cells for your B and T cells that would be complementary to that original antigen shape may no longer be complementary to the new shape. And that means that all of those memory cells that you have created within your third line of defense to defend against that original strain of the flu virus are actually ineffective and won't work against the new strain because there are different antigens on its surface. So that means that your body would then in fact have to go through your entire adaptive immune response again to create a new set of effector cells and memory cells against the new strain of the virus. And that's kind of why we are encouraged to get yearly flu vaccines, because each year scientists have studied the mutations that occur in the influenza virus and they've taken note of what proteins or antigens have mutated, and they have then designed the vaccine to incorporate those mutations so that it's going to stimulate your B and T cells to create memory cells to the new antigens that will be appearing on those flu viruses that will be spreading through the population. Another version of mutations in viruses, similar to, so we've just talked about antigenic drift, okay, where we're changing the shape of an antigen on the surface of a virus via a mutation that happens within the virus's own DNA or RNA. We're now going to have a look at a different type of change in virus structure, and that one's called antigenic shift. So this is where an individual or an organism might be infected with 2 or more viruses. Okay, so we can see on the diagram here, we've got this version of the flu virus, which is H1N2, and I can tell that because it's labeled below. And then we've got our second virus that has infected this individual, and this one's called H2N3. Unfortunately, this individual has been infected by both of these viruses, and what happens is if both of these viruses actually manage to infect the same cell at the same time, their DNA and RNA can get muddled up and mixed together. And what that means is when that infected cell releases those virus particles after the host cell has replicated the viral DNA or RNA, those virus particles that are released can kind of be a hybrid. So we can see this third virus particle down the bottom here, H1N3. It actually contains DNA or RNA from the first and the second virus. So it's a hybrid. It's able to produce the antigen H1, which came from the first virus over here, but it's also able to produce the antigens N3, which came from the second virus over here. So we've now created a hybrid virus via antigenic shift. And similar to what we were talking about before, that may mean that your memory cells and your antibodies that you had previously created to that original virus are no longer effective to this new virus. Antigenic shift is also a mechanism that scientists are particularly interested in looking at, because this is the mechanism that can sometimes take a particular virus that maybe is harmless to humans or not very dangerous and actually make it quite deadly to us. So there was a lot of talk a number of years ago about swine flu or avian flu, which are virus particles that infect obviously pigs and birds. And they were worried that if this particular strain, this avian flu or the swine flu, underwent antigenic shift, that virus would actually be able to be transmitted from a pig or a bird to a human. And because we obviously have very different immune systems to pigs and birds, it means that our immune system would really, really struggle to combat that virus, and it could potentially turn into an epidemic, worst case scenario, another pandemic. Okay, so it's kind of concerning that sometimes viruses do have this ability to undergo antigenic shift, and right now we don't have anything to prevent this from happening and creating these new super strains or hybrid strains of viruses out there right now.